Okay, good morning, everyone. It is Monday, March 30th, and this is the first online lecture. Um, this is how I will be delivering material for the rest of the course. I will be posting videos to YouTube, and you can watch them whenever you would like. You don't have to, we're not doing this all together simultaneously. So it's there for you to watch whenever you'd like, as many times as you would like. I encourage you to maybe stick to the schedule, like when we would normally meet, like try watching them at that time. Create some sort of like schedule for yourself, just so you don't get behind. So it's good to see, I don't see you, but I just wanted to say that I hope you're all doing well. I do miss seeing you in class. It's okay. I know that some of you are glad to not have seen me. I'm just kidding. I sincerely hope that you and your family and friends are doing well, and that it stays that way in the coming weeks. It's definitely a crazy time for everyone. And if you're lucky, I think the only thing you're dealing with is boredom. If at any point you get sick, someone that you know and that requires your help gets sick, let me know as soon as possible. Even if you're not sure if it's coronavirus, if it somehow impacts your ability to complete this course, I want to know and help you. So, yeah. So the majority of the lecture is not going to be looking at my face. I'm going to screen share slides with you. The slides will be available on Moodle. Hopefully, we'll see how the resolution of this turns out on YouTube. You might be able to follow along by just watching the video, but you might also have to open up PowerPoint slides and have them available. Okay, so overall, the message I want to send to you today as you're watching this from home, wherever you happen to be, is that this transition to online teaching, it doesn't make sense to rush through topics. It didn't make sense to begin with. We were already a little behind, and it really doesn't make sense to do it now that we're having to switch to online. So before I get started on today's actual lecture, I briefly wanted to go over some of the updates, announcements to how the rest of the semester is going to go. I've uploaded an updated syllabus to Moodle, and I'm going to now go through that with you very briefly. I encourage you to take a close look at it on your own, but I just wanted to go through this with you. Share screen. Okay, it's sharing. How do I get? Okay. So this is the plan for the rest of the semester, okay? As you can see on the calendar, we are definitely going to have to forego some topics that would normally be covered in this section of Biochemistry 1, namely the metabolic pathways will not be included. I was already sort of on the fence about this, and now I just I apologize to those of you who could have used the review or have never seen this before, but it's just the way it goes for this semester. So this week, March 30th through April 3rd, what I'd like to do is to get started on aspects of signal transduction. In the original syllabus, this was primarily chapter 12, and but I'm actually going to start in chapter 6, which is a chapter that we actually have already covered, but this particular section we haven't covered. The reason I'm doing this is because it deals with a signal cascade that's not talked about in chapter 12. In particular, blood clotting. So the reason why I'm spending a little bit of time just today talking about this, today and Wednesday talking about this, is that what is likely going to happen for lab is that we will still have a big cumulative final lab report. The good news is that you did all of the hard work for the most part before break with your protein purifications. I'm going to have to find a way to somehow get into the lab, and then I will just simply develop the Western blots for you and the Kumasi stain gels and send you pictures of those. So what I'm trying to say is there will still be a big lab report that each of you will have to write up and submit. It will be sort of like a standard paper with an introduction, method section, results section, and discussion section. So some of this material for the coagulation signaling pathway or blood clotting signaling pathway is discussed pretty nicely in chapter 6.5. And actually in the beginning of 6.5, I thought this presented a nice opportunity to summarize some of the things that you talked about, each of you in your groups talked about in your protein presentations, which you all did, for the most part, like a really great job of doing. Okay. So again, we covered chapter 6 through section 4 when we were talking about enzyme functions. We're going to talk a little bit more about it in the context of post-translational modifications and the cleavage of proteins and enzymes to regulate their activities. On Friday, we'll work backwards a little bit and talk about general features of signal transduction. And then we'll highlight what some of these general features were in the blood clotting pathway. And then next week, we'll talk about G protein coupled receptors. So signaling cascades that revolve primarily around these G protein coupled receptors. It's somewhat arbitrary as to why we're going to focus on these particular signal transduction pathways. It was covered last semester, but more importantly, it has some important lessons as it pertains to second messengers, which are essentially molecules that help relay signals from receptors at the cell surface to other parts within the cell, such as the nucleus, where gene expression can be altered. So this is, I think, is important for us to cover. And then there's just a lot of really interesting physiological consequences of signaling pathways that focus on G protein coupled receptors, in particular as it pertains to vision, smell or olfaction, and gustation, which I believe is taste. I know it is taste. So that's the plan for the next 2 weeks. I won't go into details right now. I won't like summarize what we'll be doing in the last 2 weeks, other than to say it's going to set what would have been set us up to talk about the details of actual metabolism, metabolic pathways. So while it's unfortunate that we won't get to talk about all of those details, some of you may be excited because it's a lot of detail. If you ever do want to remember or learn about glycolysis, the Krebs cycle, electron transport chain in great detail, having a knowledge of all of these concepts that we will still cover, such as bioenergetics and thermodynamics, chemical logic and common biochemical reactions, phosphoryl group transfers, and redox. All of these will help you jump into that much more readily. Okay. So in the last 2 weeks of class, we'll delve into chapter 13. That's the calendar. I'm not exactly sure what date the final exam is. In a lot of ways, it won't really matter because I'm likely just going to make something and post it online, and then it's going to be open book. So I'll work to figure out when exactly the official date is, but there will likely not be a time limit, and it will be open book. So whenever, as soon as I create the final exam, I'll post it on Moodle. The next part of this document is just showing you like how the grade was going to be determined initially. And then as a result of some of these changes we've had to make, it's also going to impact the point distribution. So this is initially, the first table is initially how your grades were going to be computed. And down here it's updated. And I've tried to highlight in yellow the parts that are changed. So for example, there will not be a third exam, not excluding the final exam, right? I debated between maybe we should have a third exam and then a cumulative final exam. It's gonna be crazy enough as it is. So we will only have 2 in-class exams, the exams you've already taken, exam 1 and 2, and then there will be a cumulative final that covers these topics that we're covering in the last 2 weeks. It'll be sort of half covering this and half covering what was covered for exams 1 and 2. So I kept the total points for these 2 exams at 300. So I'm going to take your scores for exams 1 and 2 and scale them to be, instead of 100 points each, 150 points each. I am almost done grading exam 2. It's sort of the same thing where I basically finished your first attempt, but attempt number 2 is taking me just longer than I had hoped it would take me. And I'll post these on Moodle as soon as possible. The cumulative final exam, it actually didn't really change that much. I just had to add on 2 points to make the total points be 800 points. So that it's roughly the same percentage of your grade. The in-class quizzes, we didn't have any quizzes this semester. Instead, I'm using this as a way to give you credit for other forms of participation, namely the midterm survey, the course evaluation that I asked you to anonymously post on Moodle. That was worth 25 points. And then I'm just now posting the actual progeria assignment. This is related to the Life According to Sam documentary, but it also includes reading a few papers related to the biochemistry of the protein that's mutated in this disease. So that will be 100 points that will be applied to your quiz grade. So I highly encourage you to start that as soon as possible. I wish that I could have posted that earlier this week, but I have been dealing indirectly with some of these, not myself, but coronavirus, unfortunately. So initially we had 7 problem sets, but now we only had 3, including the week 1 survey. I'm not going to include any more of these at this point. So this is probably the thing that was most drastically changed. It's only going to be 5% of your grade now, whereas before it was roughly 17%. Most of these points were then redistributed so that you could get them through extra credit on exams or the in-class activities. And then the last minor thing that I changed was the reading assignments that you already did. Hopefully, the reflections on the core concepts that you did at the beginning of the semester, the reflection on the Bruce Alberts review. So that, most of you who did it did an outstanding job, and I could tell it took a lot of time. So I just increased this to 70 points. So in almost all cases, it's helping your grade. And then the last thing is extra credit. The card sorting task, we brought it up at the beginning of the semester, but then never really talked about it again. At this point, if you've already done it, congratulations. You're probably well on your way to getting 10 extra credit points. If you haven't, you can try and submit it before the end of the semester, but I just decided to put that in the extra credit column. On page 2, it essentially repeats what I just talked about, but it's in bullet point format. You can see I have it divided into different sections, such as I make a list of everything that will have to be done that we have left. It includes 4 in-class activities. They will each be due Friday, I should say, or Wednesday, if Wednesday is the last day of the week, because we have Good Friday coming up by midnight. And they'll be worth a little bit more points, 6.5 points, just because I want to make sure that you're still paying attention to these lectures. I'm trying to encourage you to keep participating. The next big one is the progeria reflections. I'm going to make a different video explaining this in detail because it's worth so many points. But there will be an option for up to 10 extra credit points if you post some of your reflection answers to an online discussion forum on Moodle. There are different due dates for these. But the majority of the points will be just your final individual responses for all of these questions, which are due on April 24th. So these 2 due dates for the progeria assignments are indicated here. The initial one, which has to do with you posting your comments on the discussion forum, is due April 13th by midnight. And then just all of the answers. So this will only be some of the discussion post will only be some of the answers. This one will be all of them, and it's just your individual responses. And that will be due on the last day of class. I encourage you to finish these well before the deadline to just get it out of the way. I've tried to give you basically the full month to complete this assignment. It's a nice review of a lot of different concepts that we've talked about throughout the semester, just relating it to disease more explicitly. Okay, so we have the progeria reflections, then the final exam. These bullet points are not correct. I was going to say that half of it will be covering stuff from exams number 1 and 2, and then the other half will cover what would have been exam number 3. And this will include parts of chapters 6, just section 0.5, and chapters 12 and 13. But really, just focusing on these online lectures. Okay? The biology card sort task, at the beginning of the semester, I said you could work on it individually. In this case, you would have 16 cards and questions or questions. If you were working in groups of 2 to 3, I asked that you do twice that amount, 32 cards and questions. I can try and make a brief document reminding you of what each of these questions are supposed to be. But for now, I want to try and get to the actual lecture for today. So I'm going to leave that be for now. This you can read on your own. It's a summary of what I talked about above. Here's a more detailed explanation of the final exam. And the plan for lab, I think that, again, once I get into lab and I am able to get you some pictures, you can complete your final lab report. But you can go ahead and get a head start by writing the introduction and the methods section. Okay? So why don't we get started? I'm not sure exactly how long that took me, hopefully not too long. Give me 1 second. Okay. So what I wanted to talk about here was I wanted to bridge what we've covered thus far to signal transduction. You all did a really great job on your protein presentations, and I wanted to summarize some of the themes that you have covered before we just jump straight into signal transduction. I've tried to make this as interactive as possible. So I highly encourage you to hit the pause button whenever you see this and try to answer questions on your own. It will help you review for the final and hopefully make it a little more exciting. So this is where we will end today. This is an example of a signal transduction pathway, right? It's the one that's involved in allowing organisms that have a circulatory system to make clots in their blood. This prevents you from bleeding out, right? And you can see there's probably, you notice a few things. There's tons of arrows and they all seem to be pointing at one another. So I'd say for the final exam, really focus on this particular pathway. It's figure 641 in your textbook on page 233. We won't really discuss it much today, but we will on Wednesday in much greater detail. So this week's in-class activity will be focused on this pathway. And this is all I would like you to do. Pretend you're explaining this to a non-expert, okay? So by non-expert, I mean, say, a younger sibling, a parent, someone with minimal biochemistry knowledge. Like, they sort of know what a protein is, but that's it. So don't be afraid to get like silly, but be thorough in what you're explaining. So what you should do is, for this in-class activity, is write a few paragraphs explaining what each of the components of this pathway are. So that would be each of these little things, and how they interact with each other to cause a blood clot. So you can explain sort of how there are two different pathways and what each of these things are, and perhaps most importantly, what these arrows mean, okay? If you look really close at this, you might recognize one protein in particular, fibrinogen, right? That's one of the things that you purified, hopefully, in your in purifications of rabbit plasma in lab. So I wanted to take the time to actually explain to you what the heck is actually in plasma. and in particular, how it relates to blood clotting. So be creative, feel free to use analogies, and post this on Moodle by Friday instead of just a normal Dropbox where only I would be able to see it. I'll make it such that you can see your peers' responses after you submit. Feel free to reply to some of your peers. You know, I know like we're all like, I don't know, at home, maybe kind of lonely. I just want to give you as many opportunities to interact with each other as possible. For fun, we'll vote via Moodle poll for who has the best response. And the top three voted will get 5 extra credit points added to your final exam score. So it has to be a combination of creative but actually good scientifically, okay? It should really, yeah. It can't just be funny and cute. Anyways, okay, so big picture, right? We've spent a large amount of time studying one enzyme, right? Alcohol dehydrogenase, pepsin, what is it, retinoid isomerase. Anyways, you know the names because you spent so much time studying them. We spent all this time studying just one enzyme, its catalytic mechanisms, as well as its regulation. But the thing that I want you to appreciate is that in the context of how these enzymes actually mediate cellular phenomenon, which is inherently complex, these are things that we really care about when it comes to understanding how disease occurs. So this is the bio of biochem. What you need to realize is that it is a group of enzymes that work together to carry out processes. So basically, you've done step 1 of being a diligent biochemist. Now we need to do step 2 and realize that it's a hell of a lot more complicated than just that. Everything is connected, and the signaling cascades is really a way of looking at biochemical processes more holistically. Where's Waldo is a fun thing to do. For those of you who aren't familiar with it, you are shown big pictures with lots of different people, and you have to find where Waldo is. This is really very much what signaling transduction is, except we're dealing with where is your enzyme in the context of a very crowded cell, right? So, no cheating, okay? Hit pause and find Waldo. I have no idea if this is gonna work or not because I have no idea if the resolution of this is good enough, but go ahead and hit pause. What I'm gonna do is I'm just gonna say hit pause and trust that you hit pause. Can you find Waldo? That's what Waldo looks like. Okay, welcome back. Who found Waldo? This is really weird to make this video like this. I just feel really weird. That's where Waldo was. Who found Waldo? Oh, so that's a zoom in of Waldo. You know, looking at this picture, I don't want to get too off topic, but I think I see Waldo here too. So maybe there's 2 Waldos. But we have to stay on track, okay? We don't have time anymore. This is a zoom in of what is clearly Waldo. And a signal transduction pathway is like saying, I don't know, let's say like Tina and Jennifer did a nice job talking about Sarc kinase, right? So this would be like Sarc. But now we want to understand how Sarc is related in the context of a really complicated cell signaling pathway. And so you'll start to see arrows drawn between individual members that describe different interactions and or modifications. Oh, I was trying to pretend that's, I don't know. Congratulations, Jaden and Ayan. I thought maybe you guys found Waldo. Oh, by the way, Jaden, hopefully you're watching this. Everyone, well, this isn't being done live, so it doesn't really matter, but I guess say hi to Jaden. Unfortunately, he had to go back home. Usually at this point in the, usually when a foreign exchange student leaves, I like to say bye to them as a class. But unfortunately, we weren't able to do that. So I just thought I'd say we miss you. I mean, everyone misses everyone because we can't see each other, but you're not here in the United States anymore. And I wish we could have given you a proper farewell. Um, we hope that you and your family are well. Okay, so where's Waldo? Not where's Waldo, but where is a protein or enzyme that we're interested in studying? This is an example of a signal cascade. It's called the EGFR signaling pathway. EGFR stands for anyone? Wow, good job. Epidermal growth factor receptor. Yeah, it's right there. If you open up the PowerPoint slides and click on this link, it's actually a really nice somewhat interactive picture from a biotech company called Biorad. You can zoom in and out and also look at a figure legend. I'll just say that this pathway is really important for things like cell proliferation and migration. When this pathway goes awry, when these cellular processes called cell proliferation and migration, when these are not correctly tuned in to be coordinated with a signal, it leads to cancer. Okay, so let's play this game with with Sark. Which one group presented on? Tina and Jennifer. No cheating. Hit pause. Find Sark. Okay, welcome back. I hope you found Sark. You see it? Bam. There it is. There's a C in front of it. I don't know what the C stands for, but it's Sark. Okay? Maybe I should clarify what's actually happening here. This is the plasma membrane. This is the outside of a cell, and this is the cytoplasm. This is the nucleus. Okay? So it's all about transmitting a signal from the outside of a cell to the interior of the cell, sometimes also not only transversing the plasma membrane, but also the nuclear membrane. Okay, so this is, next slide is just a zoom in of where Sark was. And now I'll ask you another question. What is Sark interacting with in this cascade? Pause. Okay, hopefully you saw that if you look at Sark, there are arrows going to at least two things. This is highly simplified, but in the context of studying things like cell migration and cell proliferation, these are the relevant proteins that we deal with. So Sark is directly interacting with this receptor that sort of is where the whole cascade pathway begins, as well as something called SHC. So you can see there are at least two different binding partners here. So what do these arrows and green symbols mean? What do you think that they mean? Here is a picture of the legend. If it's too small, go back a few slides and click on the link and take a peek at these. But try and tell me what types of molecules Sark, SHC, and EGF are, as well as what the interactions are representing. Okay, hit pause. All right, welcome back. I accidentally already had one of the boxes highlighted. So there are essentially two sets of answers here. The first is that Sark is a kinase that adds phosphate groups to proteins, phosphorylates them. And this first arrow here is just showing you that it phosphorylates a generic binding protein called SHC. So its function is, SHC's function is primarily just to bind to other things in this signaling pathway. The second answer, if you listen closely to Tina and Jennifer, we hopefully appreciate that a kinase, so it's pronounced kinase, is something that phosphorylates other things. So to SHC, its function was primarily to phosphorylate it. You can see that SARC, there are different types of kinases. Some phosphorylate other proteins, others phosphorylate other lipids. SARC is actually a protein kinase, and it is, in the case of the receptor, it's not only phosphorylating, but sometimes when it simply just binds to the EGFR receptor, it also can modulate EGFR's activity. EGFR itself is a receptor, not just a generic receptor, but a receptor with enzyme activity. So it itself can catalyze chemical reactions. Good job hitting pause and thinking. If you didn't, seriously, I encourage you to at least try. Not shame on you, but please just try. So a potential exam for the exam question for the final, I might provide you with a link to some random signaling cascade, similar to the EGFR, apologies for the typo, signaling pathway that for the link that's provided to you. So I do encourage you to visit this, try and explore it, and make sure you can describe what's actually happening. I might ask you to explain certain arrows to me or apply this knowledge to some type of imaginary scenario. So go back to slide 4 and click on that link. So simple review question, what type of modification is phosphorylation? Non-covalent or covalent? And of those, reversible or irreversible? I'll hit pause here. Okay, hopefully that you got this answer. It's a covalent modification, so there's a covalent bond made, but it is reversible. Okay, this is leading into the part of chapter 6, section 5 that is discussing, so I'm on page 225, and parts of 226. The question here is, how are enzymes' activities regulated? This is one way in which they are regulated, through covalent reversible interactions. Most of the enzymes in pathways follow kinetic patterns that we have already discussed in chapter 6. This is Michaelis-Menten kinetics, right? However, while this is important in some contexts, Michaelis-Menten or enzyme kinetics in general, this is important for things like drug design. As you have seen in the previous slides with signal transduction cascades, simply knowing the kinetics of a single enzyme is usually not enough to predict a physiological outcome. Why? Because there are several other enzymes, either upstream or downstream, of your enzyme of interest. Moreover, not all enzymes are equal within a given signal cascade. Some impact the overall physiological process more than others in different situations. This dynamic behavior in biochemistry is what gives rise to the dynamic behaviors studied in courses you may have taken before, like cell biology. So this is, for example, this is a really neat example of a physiological process. Um, it's easy to appreciate the bigger up you get, I think, especially when you start to see how cells behave. It's easy to appreciate how biology is dynamic. But as you're watching this video, think to yourself, this isn't just random, it's highly regulated. And what this all comes back down to is the highly regulated nature of individual enzymes within signaling pathways. So this is a movie of actually what you're going to see flying around is an immune cell that's migrating in the inner ear of a zebrafish, of all things. The point is, as you watch part of this, is why the cell is moving, and it is actually moving in a coordinated way. The answer is going to be that there are signals, the absence or presence of signals that lead to differential activation of signaling cascades. So this thing right here is actually, all these things moving are immune cells. Let me fast forward to slightly later. It actually shows you the trajectory. So you can see they're keeping a line to show where the cell is moving at different parts. The idea is that the immune cell is a surveillance tool. It's looking for potentially damaged cells and it may trigger a defense mechanism, primarily the inflammatory response, to recruit other immune cells to come and kill it. Okay? But again, this is all coordinated. And you have to impact, it starts at EGFR, but it somehow has to come down, and in this case, cause a change in gene expression that leads to some cell physiological process. In this case, that is the migration of cells. So STAT3 and STAT1, I just thought I'd mention, this is one of the more downstream parts of the pathway. What are they? They're inside of the nucleus. Here. So can you see these shapes? Again, I just want you to be able to look at these signaling pathways and, given a legend, be able to say something intelligent. But STAT3 and STAT1 are what? As you can see, if you were to look closely enough, they're actually transcription factors, right? So we started at the plasma membrane where there's a receptor. A bunch of different things are binding B to it. The binding of that causes binding of other things. In some cases, it causes phosphorylation of things. There are different branches of this pathway. One of those branches, if we head down the JAK1, JAK2, these are also protein kinases. This is a separate arm of then SARC, which was discussed in class. If you go down this route, you actually activate two transcription factors, STAT1 and STAT3. Hopefully you remember from cell biology or even introductory biology that transcription factors, what do? They bind to promoter regions of genes and transcribe the DNA into RNA, which can then be made into protein. All of this has to happen in the context of a crazy cell moving like this, okay? So it's just kind of crazy to think that this might be sped up a little bit. I don't know if maybe this is seconds or minutes. But all of these things are happening when a cell is moving. Not just binding of something to a receptor, but also all of these other different things leading to, in this case, activation of gene expression in the nucleus. More proteins are made that ultimately help the cell migrate. So these two lines here, although they're just showing you two simple lines, each transcription factor leads to gene expression, which then, you know, it's very complicated and it happens fairly rapidly. Okay. So if we move back to the textbook quickly, some enzymes, right, are called regulatory enzymes. This is just used to describe those enzymes within a huge signal transduction pathway, those that have a supposed greater effect on the overall sequence, whether or not this pathway is activated or not. What makes them unique is not just their importance in mediating the rates of chemical reactions, but more importantly, they are sensitive to signals, okay? Signals can be a lot of different things. Can any of you think of an example of a signal, right, that might activate a part of a signal pathway? It may have activated or inhibited your enzyme of interest. I want you to be able to describe some signals that originate from outside of the cell or from inside of the cell. So go ahead and hit pause and try and maybe on your notebook, jot down a few examples. Pause. Okay, welcome back. I wish we could share them as a class. There's lots of different ones to choose from. These were two that I chose. So signals that originate from outside of a cell. This could be if you think of neurotransmitters. These are short-range signaling molecules that bind to receptor on the membrane of neurons. Right? This signal must be transmitted to the nucleus, leading to a change in gene expression. So similar to the example of the EGFR signaling pathway, right? We have to go through a receptor at the plasma membrane. And this can lead to post-translational modifications such as phosphorylation that ultimately lead to a change in gene expression. Signals from within the cell are also really important. The most obvious example is, at least to me, DNA damage, right? So as a cell, you not only need to know what's happening outside in your environment, you have to be aware of critical events that are happening within the cell. So when there's DNA damage, it triggers a signaling pathway that recruits DNA damage repair mechanisms, right? So at this point, I guess the million-dollar question is this: how are the activities of regulatory enzymes actually modulated? Okay. In a lot of ways, this is review. You talked about these in your protein presentations, but let's just bring it all together real quick one more time. So think about not only your own protein presentation, but other people's protein presentations. Can you think of different ways? Pause. Okay. So in some cases, we had reversible non-covalent binding of compounds, right? These are called can typically be called allosteric regulators. These are comprised of things like small metabolites or even cofactors. Okay. The important part is that it's reversible. Here it's also reversible, except we're dealing with covalent addition of chemical groups, which was that little question I had earlier. Phosphorylation would be an example of this. So you can add a phosphate group, but you can also remove a phosphate group. Out of curiosity, do you know the name of the enzyme that removes phosphate groups? We know that those that add phosphate groups are called kinases. Those that remove are called phosphatases. Okay. There are many different types of post-translational modifications. We'll discuss some briefly in a second here. So even though these are different, how are both similar in terms of how they regulate activity of enzymes, how they modulate the activity of regulatory enzymes? Maybe take another quick second and pause. Think structure-function relationships. So the answer is that they cause a change in shape, right? But go one step further. So here I've said they modulate activity by causing conformational change. It's true in both cases. But why does a conformational change actually cause the enzyme's activity to be changed? What does this conformational change do? So there are at least two answers, right? It can reveal or hide a substrate binding site, or it can directly impact enzyme catalysis. All right. So these are two examples of how activities of regulatory enzymes are modulated. Can anyone think of another? I'm looking at you, Kao and Faith. Kao and Faith talked about pepsin. And how was pepsin's activity changed? It started in an inactive state and was then activated by, we were missing one, proteolytic cleavage is the final way, okay? So here are the three different ways that you all did a great job of talking about. In the case of, it's a very common way for proteases, which are enzymes that degrade other proteins. Proteases are typically activated by this mechanism. Something cleaves them, converting them from their precursor form to their mature form. Here, a covalent bond is removed, whereas here it's added. Because the covalent bond is removed and then whatever was on it just is sort of floating in space, oftentimes this is irreversible, okay? In terms of how the removal of something can impact its activity, oftentimes it can cause an overall conformational change in whatever is left over. But more commonly, what was cleaved off can actually be something that the enzyme uses to bind to someone else, right? So it could go the other way. It could inactivate a regulatory enzyme in addition to activating. In each of these cases, simply because something is post-translationally modified, it's hard to know whether or not the enzyme's activity is increased or decreased. So here we're going to focus on the reversible covalent addition of chemical groups or the irreversible proteolytic cleavage, the removal of a covalent bond. In terms of what to focus on in the textbook, if you were to go, you can skip these two sections on pages 226 and 227, right? I want to get to, if you were to start back up, I would read page 228, the section that says some enzymes are regulated by reversible covalent modification. It mentions a lot of different types of reversible covalent modifications. Some of them are included here, right? For some of them, we've talked about adding a phosphoryl group or phosphate group. acetyl, apologies for the typo, carboxyl groups. The point is that a lot of these are relatively small and simple, and the main thing I want you to appreciate is that they are not always small and simple. By simple, I mean by biochemical standards. You can also have the addition of things like sumo through a process called sumolation, or ubiquitin through a process of ubiquitination. These are particularly important for what biological process? The answer is protein degradation. So there are multiple ways in which proteins are targeted for destruction in a cell. Sometimes they're damaged, but in order to be properly degraded, an enzyme comes along and actually attaches an entire protein to the protein that is to be degraded. What do I mean by that? So sumo and ubiquitin, these are the things that are covalently attached to a target protein. They themselves are proteins. So they're huge. You're adding an entire protein to another protein. The other thing I would like you to focus on reading independently is on page 1111. All right. You'll see a section on this page that says addition of word corrected, or this automatically corrected, isoprenyl. Don't correct that. Isoprenyl groups. This involves a type of post-translational modification or reversible covalent modification called farnesylation. The reason why I'm pointing this out to you in the textbook is because it's really important for the pathology of progeria, which is what the documentary and the additional papers that I'm posting deal with. So here it's actually adding in something called a farnesyl group. It's somewhere in between here and here in terms of its size and complexity. But in addition to the in-class activity, where you're working on explaining the blood clotting cascade to a non-expert, think about this question, and I'll try and start with this on Wednesday, right before we get into the nitty-gritty details of blood clotting. What does farnesylation do to the protein that it's being attached to? It's really important to be able to answer this question to complete the progeria activity. Okay, and see, here I made it huge for you because I thought it was important. What does farnesylation do to the protein that it was attached to? In particular, here, I'll give you a hint. It does something to this protein's cellular localization. What do I mean by cellular localization? The cell is spatially organized. Within the eukaryotic cell, at least, you have different organelles, right? You also have different areas within the cytoplasm. It's important not only to regulate an enzyme's activity, but its localization. Farnesylation helps certain proteins localize to something very specific. See if you can figure that out. Okay, if we move on real quick, because I'm sure I'm running out of time, pages 229 and 230, you can skip. We will talk about phosphorylation in chapter 13 in a little bit more detail. For now, we can go ahead and skip this. And this is where I want you to start for Wednesday. At the bottom of page 230, there's a section that says some enzymes and other proteins are regulated by proteolytic cleavage. So go ahead and read this section. For people like Cal and Faith, it's going to be review because it's talking initially about proteases and how protease activity is regulated by cleavage. But then it's quickly going to lead into the idea that proteases aren't the only regulatory enzymes whose activity is modulated by cleavage. There are also others, namely those involved in blood clotting. If we go back to this initial thought experiment or this thought activity I had for you, where I asked you to think about signals and signaling cascades where the signal originates outside of the cell and is brought in versus those that originate inside of the cell. Blood clotting is sort of this weird thing where it's actually the signal is originating outside of the cell, and the regulatory enzymes that are important for blood clotting are outside of the cell. So it's like an example of an outside is how I kind of think about it. So the signal itself, which would be like a cut, some part of your circulatory system has been damaged. That's the signal. But the enzymes that need to be activated are also outside of the cell, which is kind of cool. So there's no need to have a signal transmitted across a membrane. I think that's pretty neat. For fun, here's figure 6-40 from your textbook on page 232. You know, you've spent a ton of time pipetting clear liquids into other maybe less clear, like crap is floating around in them, columns, and then spinning them. I'm just trying to describe how you may feel protein purifications occur based on your experiences in lab. It's kind of nice to be able to think about what those things were by looking at real pictures within a cell. So this is a picture of fibrin, which is related to fibrinogen, which is one of the fractions you try to purify. And I thought it would be nice for you to maybe just be able to look at this picture and identify the different components of the blood clotting system. So here, there are actually three things shown. There's the blood clot itself, which consists of aggregated platelets. The second thing is cross-linked fibrin, which is a protein that is initially soluble, but then starts to aggregate. And then the third thing is erythrocytes. So can you identify these three things? You may not have much of a biology background. So some hints are the platelets, they're relatively small blood cells relative to erythrocytes, which are larger and they look like discs. And then, as I said before, fibrin is a protein that aggregates to form thin filaments. So can you identify them? Pause. Okay, welcome back. The platelets are smaller, and these are actually indicated here and here. So they're the sort of light colored yellow things. They're tiny blood cells that are cross-linked by this network of meshy stuff, which is in number 2 here, the cross-linked fibrin. And then here are your huge blood cells, and some of them you can kind of see they're disc-shaped. But number 3 is actually the erythrocytes. Okay. This is a schematic of fibrin. So you can see it has many different subunits. The alpha subunit, the beta subunit, and the gamma subunit, right? So it is an oligomer. We've talked about dimers and tetramers, dimers 2 and tetramers 4. These are examples of quaternary structure. So I'll wait until Wednesday because I think I've run out of time to discuss this in more detail. But this is really going to help, I think, with making sense of what it is you were purifying in the protein purifications. So take a peek at this figure as well and try and explain what's going on here. The challenge is to be able to explain why when you cleave something, it now starts to form long chains, essentially aggregate. This aggregation is important for forming these filaments that essentially stop the flow of blood. And then, as I said, this is where we would end today, section 6, or figure 6-41. So go ahead and try and explain what each of the, what's actually happening in this figure to a non-expert. Okay, well, that marks, I believe, the end. Zoom. All right, well, that marks the end of this first lecture. I apologize if it was a little whatever, but take a peek at some of the things I said to work on, and I'll see you on Wednesday. Thanks.